Hello everyone and welcome to this presentation. I'm Gaurav Agrawal, Scientific Development Manager at Eurofins DiscoverX. My role here is to support scientific integration of our cell-based assay platform in drug discovery programs globally and thereby building strong partnerships with biopharma industry. So thank you for joining me today. The presentation today is mainly focused on how our qualified cell-based assays can accelerate the development of therapeutics targeting pro-inflammatory cytokines that are implicated in COVID-19. Now, with over 23 million cases and 800,000 total deaths, the fast-growing pace of COVID-19 pandemic has been challenging the capacities of healthcare systems worldwide. As we have seen, the pathology of COVID-19 in severe cases shows an aggressive immune response, and for that reason, there are several ongoing clinical programs that are evaluating the therapeutics that are targeting the inflammatory signals as a treatment strategy for COVID-19. Now, the high mortality rate in COVID-19 patients has been linked to cytokine release syndrome, commonly referred to as cytokine storm, which is an excessive and dysregulated release of pro-inflammatory cytokines by body's immune system. This leads to an uncontrolled inflammation leading to acute respiratory distress syndrome or ARDS. This is evident from highly elevated cytokine levels in patients with ARDS. Here I have put together a quick recap to pro-inflammatory cytokine pathogenesis in COVID-19. The initial point of contact with SARS-CoV-2, as we know, is the ACE2 receptor on the alveolar cells. Once the virus infects the alveolar cells, they trigger an innate immune response in the host, where the immune system in the lungs or immune cells in the lungs releases inflammatory signals like interleukin 6 and other chemokines to attract other immune cells to the infected tissue. Now from here, it can go two ways. First and most common, where things go as expected and the immune response by the host is mounted proportionally. The infected cells are cleared by CD8 positive T cells and macrophages, followed by B cell activation, producing antibodies, which helps in clearing the virus. But it can also go in a different direction, where the body mounts a dysfunctional immune response to the infection. This is mainly caused by cytokine storm, where pro-inflammatory cytokines such as interleukin 6, interleukin 1 beta, TNF alpha, GM-CSF are released by the immune cells. In fact, the levels of these cytokines are very high in COVID-19 patients that are in ICU. These cytokines cause acute inflammation of lungs and can lead to death due to ARDS in severe cases. Now it is still unclear and it's an active area of investigation as to what factors make the immune response go one way or the other. But in broad terms, these factors are associated with age, smoking status, or pre-existing conditions like heart, lung, or kidney disease, and even diabetes or cancer may play an important role in disease progression. So what we are doing about combating cytokine storm. Here I have listed the key cytokines and chemokines that are mainly implicated in cytokine storm with their respective therapeutic drugs that are currently in clinical trials, such as Tocilizumab targeting IL-6, Anakinra targeting interleukin 1, and Adalimumab a neutralizing antibody for TNF-alpha, where each of them are in several clinical trials across the globe. The figure mostly lists repurposed drugs here, but there are also many new therapeutics such as the ones listed for GM-CSF like Adalimumab and IL-8. Now for development and characterization of these therapeutics as well as releasing them into the clinic, reliable functional assays are required. And this is where Eurofins DiscoverX can partner with you with our reliable cell-based assay portfolio. We have functional cell-based assays for each of these cytokines and chemokines. And most importantly, we have qualified and MOA-based bioassays for all of these cytokines. These bioassays can really accelerate your drug development and release programs, and how they can do that would be the next part of the presentation. So in this part, I will introduce you to our cell-based assay platform and present a case study for Tocilizumab bioassay, which is designed for potency and stability testing for therapeutics targeting interleukin 6. With that, I will give a quick overview of bioassays with 3 other relevant targets. As you can see here, these bioassays have been qualified with the marketed drug, which makes them highly relevant for biosimilar and biobetter drug development. Following this, I will discuss our signaling reporter assays, which provide a more downstream readout. And towards the end of the talk, I will tie it with another set of assays for characterizing small molecule drug candidates and discuss functional assays for evaluating JAK1, 2, and 3 inhibitors. But let me first briefly introduce our enzyme fragment complementation or EFC technology, which is our core technology behind these assays. Now, our assays utilizes this proprietary technology, which is based on a split beta-galactosidase system, where the enzyme is split in 2 inactive fragments called ED or enzyme donor and the EA or the enzyme acceptor. When these 2 fragments are put together, they complement each other to form a functional beta-gal enzyme. We add a chemiluminescent substrate in the assay that this active functional enzyme hydrolyzes to produce a light signal that can be read with any benchtop luminometer found in most labs. EFC technology also provides a great deal of flexibility, allowing us to use it innovatively in building our versatile and robust cell-based assay platforms. Let me show how we use this technology with an example of a receptor dimerization assay for interleukin 1. Here the interleukin 1 receptor and its co-receptor dimerizes as a result of interleukin 1. It captures the physiological MOA of interleukin 1 signaling. As you will see, the receptor units are tagged with beta-gal fragments as described on the previous slide. Now in the presence of interleukin 1, the receptor units dimerize, thus bringing together the beta-gal fragments, and in presence of the substrate produces a quantitative light signal. The EFC assays are highly quantitative and typically produces a gain of signal response. Now these assays can also be utilized for quantifying an inhibitory response from an anti-receptor or an anti-ligand antibody or molecules like Anakinra in this case. And these antagonists also produces a highly quantitative response like the agonist does. And likewise, we have successfully utilized EFC technology for many other assay formats, such as to quantitate protein-protein interactions, protein turnover, and degradation, for evaluating downstream responses, and more recently, for developing cytotoxicity assays such as ADCC and ADCP. For our cell-based assays, we offer phase-appropriate assay formats. One is, of course, the stable cell lines offering a continuous culture format. But more importantly, we have the bioassays, which are our ready-to-use assay formats and don't require any cell culture. It is our highly optimized, these are our highly optimized assay formats developed particularly for potency and stability testing and are perfect for implementing in QC lot release programs. We took a step forward and qualified our bioassays with marketed drugs where available. Before I move forward, let me give you a very quick overview of assay protocol for our cell-based assays. It starts with simply plating the cells in the assay plates, usually for overnight. Next day, the molecule of interest is added and incubated for another 1 to 3 hours. Thereafter, you will add a detection reagent with a beta-gal substrate in it and incubate the plate in dark for an hour to 3 hours. That is about it. After the incubation, the plate can be read in any benchtop luminometer. The schematic on the lower right side shows a bioassay kit that comes with all the necessary reagents included, tried and tested, with a certificate of analysis document that comes with each kit. We have included everything that you will require in the bioassay kit, so there is no need for you to source any of the reagents from any third-party vendor. This way, you will be able to root out any variability that is introduced from any other non-optimized assay reagent. Perfect. I think we are ready to review a case study for one of our qualified bioassay. I have selected our Tocilizumab bioassay since it is for interleukin 6 targeting therapeutics and thus most relevant to COVID-19. The intent of this case study is to provide you with an overview on how we go about qualifying our bioassays to make them most suitable for potency and stability testing for drug candidates when implemented in a lot release program. So let's look at the assay design for this bioassay. Of course, it is based on our EFC technology and utilizes the dimerization platform, very much like the interleukin 1 assay that I described above. In presence of interleukin 6, the receptor units dimerizes, bringing the receptor units together and thereby complementing the EFC fragments. Adding the beta-gal substrate produces a quantitative gain of function signal. However, when the assay is run in presence of tocilizumab, an IL-6 receptor antibody, the binding of interleukin 6 is inhibited in a dose-dependent manner. It also produces a quantitative curve, but it's an inhibition curve. Okay, so here are some of the key assay parameters we test during assay qualification. Firstly, we test assay consistency between several dose response curves. This gives a very good idea on any variability associated with the assay. Second, we look at the plate uniformity across the entire plate. This provides us with any plate associated bias and possible edge effects. Next, we look at assay repeatability and for a consistency in the slope of the curve to ensure there are enough points in the linear range of the curve. And lastly, perform assay suitability for relative potency analysis. We determine assay accuracy, precision, and linearity over a range of 50 to 150%, and this is performed by two individual operators. After that, we perform a parallel line analysis using PLA software or Softmax Pro, analyzing the relative potency data. Here is the data for testing intra-day plate-to-plate variability. 3 experimental replicates for tocilizumab were prepared at 11-point dose response curves and run on the same plate. Similar experiments were repeated again to obtain data from 3 individual days for inter-day variability. As you can see, the curves are very uniform in these experiments. We obtained an inter-plate precision that was within 8% for the signal to background, and as you can see here, was below 10% for IC50. Also, the inter-day variability was found to be less than 25%, which is well within the acceptable range. Next, we performed relative potency experiments and parallel line analysis with tocilizumab across a range of 50 to 150%. The experiment here shows data from 2 individual analysts. The blue curve is with 100% reference, while the green is the sample. As we move from 50% to 150%, the green curve shifts towards the left, moving across the 100% reference, depicting an increase in potency. Here is the statistical analysis of the relative potency data. The assay was determined to be very accurate and precise. The accuracy was 105% and overall assay inaccuracy was within 9.3%. A high degree of assay accuracy here suggests that the bioassay can measure relative potency with high confidence. Dilution linearity was analyzed by plotting measured against expected relative potencies for each point, as shown in the figure on the right. The R-square value came just below 99.3%, showing an exceptional assay linearity. These datasets that I discussed here for Tocilizumab bioassay is a part of our standard assay qualification for all our bioassays. With such amount of assay qualification work behind them, you can be confident that when implementing them in a quality control space for your biologic or a biosimilar drug candidate. Next, I will discuss 3 other bioassays briefly and not go over the entire qualification work, but please feel free to reach out if you would like to review any of them. The next bioassay that I will discuss is for Anakinra. I did went over the working of this bioassay in the beginning, but to quickly recapitulate, the bioassay design, like all our bioassays, is MOA-reflective. The assay is used to quantify an inhibitory response from Anakinra, which is a slightly modified form of naturally occurring interleukin-1 receptor inhibitor. As shown in the bottom panel, in presence of Anakinra, there is a dose-dependent inhibition of interleukin-1 receptor dimerization. So let's look at the functional data. In blue dose response curve here is for interleukin-1 beta, with purple for Anakinra. The assay, as you can see, produces a robust window with sensitive detection and a dose response curve for Anakinra as well as for IL-1 beta. We further performed assay qualification work as I showed before for Tocilizumab. Bioassay data that is showed here for dilutional linearity was derived from the relative potency experiments performed over a range of 50 to 150%. As you can see, the relative potency data derived from the bioassay was highly accurate with over 99.4% recovery and also was highly precise. Next is our bioassay for GM-CSF, which was qualified with marketed drug Leukin. Leukin is a recombinant form of Sargramostim produced in yeast. Both Sargramostim and inhibitory antibodies against CSF2R, such as Adalimumab, are in clinical trials for COVID-19. Similar to Tocilizumab and Anakinra, this bioassay is also based on receptor dimerization. So let's quickly look at the qualification data here with Leukin. As you can see, this is one of our best performing bioassay with dilutional linearity over 99.5%. Also, the relative potency data derived from the bioassay was highly accurate, showing that the assay is suitable for implementing in quality control programs. The last bioassay we'll review would be the Adalimumab bioassay targeting TNF-alpha. As we know, TNF-alpha signaling goes through NF-kappa B pathway. The TNF-induced phosphorylation of I-kappa B makes it a target for proteasomal degradation. In this assay, we tagged I-kappa B protein with the EFC tag to monitor its degradation status. The level of I-kappa B protein is inversely proportional to the TNF-alpha concentration, and thus it forms a quantitative loss of signal assay. Now, in presence of TNF-alpha blocking antibody like Adalimumab, the I-kappa B protein is not phosphorylated or degraded, thus restoring I-kappa B levels in the cell and hence produces a gain of function signal. Here is the functional data. In blue is the dose response curve for TNF-alpha and in purple for Humira. The assay produces a robust window with sensitive detection and a dose-dependent response to both TNF-alpha and Humira. The assay qualification data is here with Humira. Also aligns very well, producing an almost perfect dilutional linearity with 99.9% and with excellent accuracy and assay precision. Now let's put things in perspective. Here's a typical roadmap for developing a fit-for-purpose bioassay for a biologic drug, which is needed to release it into the market for clinical use. It can take upwards of 12 months if starting from cell line development. Now this time frame is significantly reduced by more than 9 months when you implement one of our qualified bioassays. So here you can right away transfer our bioassay to a CRO site where they can start the assay validation work. This way our bioassay significantly accelerates time to market for therapeutic drug candidates and helps save significant cost and resources. Next, I would like to introduce quickly our new set of reporter assays designed to provide a more downstream readout of activation of inhibition, readout of activation or inhibition of a signaling pathway. These would be highly complementary to our bioassays that are more focused on recording upstream events. I will show you here two datasets for reporter assays focused on cytokines relevant to COVID-19. As I showed you with the bioassays. The first is the HepG2 cell background exploiting the STAT3 pathway, whose role is well documented for cytokine signaling, including interleukin 6. The figure shows a robust and sensitive response to interleukin 6 and a good inhibition curve for Tocilizumab with a large signal to background window for both. Next is our NF-kappa B reporter assay in G2OS cells for TNF-alpha. TNF-alpha, like other of its family members, signal through NF-kappa B pathway as well. The figure shows a robust and a sensitive response to TNF-alpha and a good inhibition curve for Humira with a very large over 50-fold assay window for both. Thus, combined with the bioassays, these signaling reporter assays will provide a comprehensive understanding for the drug molecule's MOA. In this last section, I would also like to highlight our functional cell-based assays for JAK inhibitors. JAK inhibitors have become an active area of investigation with several ongoing clinical trials for COVID-19. The data on the right is a functional assay where JAK1 is expressed as a cytosolic tyrosine kinase along with CSF3 receptor. When G-CSF is added, CSF3 receptor is activated and is phosphorylated by JAK1. Similarly, the assay in the middle expresses TYK2 with growth hormone and at the corner expresses a JAK1 with prolactin receptor in the JAK3 activity assay. Together in these assays, we have tested several JAK inhibitors. Most relevant among them are shown here, such as ruxolitinib, which is under clinical trials for COVID-19. I'll stop here and would like to quickly mention that we have a strong custom assay development team who can build assays or modify existing ones to suit your needs. We can also take up pilot studies if you would like to run a quick feasibility study with any of our assays. Plus, our Eurofins Discovery Partner Labs can run any assays that you see here as a full service. So, quick set of conclusions. In this talk, we have discussed our target-specific, qualified, MOA-based functional assays for drugs targeting pro-inflammatory cytokines, all of which are implicated in COVID-19 pathology. With that, we offer complete solutions, including custom assay development service. One relevant example is an ongoing custom assay development project for ACE2 receptor that would be intended for characterizing therapeutics aimed at preventing coronavirus from attaching to the alveolar cells in the lungs. With that, I will thank you for joining me today. Have a good day and stay safe, everyone.